Hi, my name is Sadie Kenny.
And my name is Suraj Patel, and the topic of our research is tracking the effects of plant growth promoting rhizobacteria. From our experiment, we found that the bacteria we collected had some positive effects on the tested plants. This is important because it provides an alternative to chemical exposure in agriculture. Oftentimes, rhizobacteria have plant growth promoting properties, or PGPR for short. The plant we extracted our bacteria from, called Scholzia californica, more commonly known as the California poppy, is native to the North American West Coast. This specific plant that we used did not have nodules on its root, and it was grown in soil with a 0.069% moisture content and a pH of 6.43. Finally, the plants that we tested the effects of the extracted bacteria from was Pisum sativum, more commonly known as sweet pea. The objective of our experiment is to investigate rhizobacteria extracted from the rhizosphere present around the California poppy root and its biochemical properties, PGPR mechanisms, and their genetic information.
We did biochemical property assays to test for the ability of the extracted rhizobacteria to produce cellulase or polysaccharides, solubilized phosphate, or fixed nitrogen. Isolates 9 and 12 tested positive for the most properties from all the bacterial strains we isolated from the California poppy, so we chose to investigate these further. We identified these isolates by extracting the DNA through PCR amplification on the 16S ribosomal DNA. We also wanted to see how our chosen microorganisms would have a practical effect on the growth of plants, specifically the sweet pea. So we did this by inoculating hydroponic boxes with our chosen isolates. We used saline as a control. We allowed time for growth, and then we analyzed the shoot and root systems. So on to our results for the biochemical properties. Table 1 shows the biochemical properties of bacterial strains 9 and 12. In the table, it shows that isolate 9 was non-determinate for ACC deaminase activity, while it was positive for all of the other tested biochemical properties. While isolate 12 only tested positive for phosphate solubilization, ACC deaminase, and exopolysaccharide production. Our results for gene sequence analysis, figure 1 shows a phylogenetic tree with our 2 chosen rhizobacteria. Based on our gene sequence analysis, the bacterial strain 9 has a 99.55 identity similarity to the genus Paraburkholderia, while isolate 12 has a 97.42 identity similarity to the genus Achromobacter.
Moving on to the hydroponic boxes in our experiment, figure 2 shows the results from our hydroponic box experiment. As shown in the graph, there was no significant difference in any of the varying root and shoot system weights. Statistical analysis showed us that the p-value of for a root system was 0.629 and 0.204 for a shoot system. Figure 3 is also a graph that instead represents the average length of the varying shoot systems. From this graph and from statistical tests, we found a p-value of 0.0106, showing us that there was a statistical difference in growth in the presence of our isolates. Finally, figure 4 is a series of 3 images featuring each sweet pea hydroponic box. The left is the control variable, and the following 2 images are inoculated with isolates 9 and 12 respectively.
So on to the discussion of our results. Again, for our biochemical properties, based on the tests performed, Paraburkholderia tested positive for at least 4 biochemical properties, while Achromobacter tested positive for 3 biochemical properties. And the results of our biochemical properties of the isolates were consistent throughout all of our testing, as well as throughout literature. In terms of the gene sequencing analysis, the genetic sequencing allowed us to match our bacterial isolates with the bacterial genus it's most closely related to. For both of our isolates, we were able to identify it. We were unable to identify it down to the species level, but our genus that was identified made sense based on found literature, as they were both consistent in description and biochemical properties.
Based on the statistical tests taken, the dry weight of the sweet pea inoculated with the bacterial strains has no significant difference from the uninoculated sweet pea plant. Statistical tests also show that the shoot length of each inoculated sweet pea is significantly different from the uninoculated sweet pea plant. We have a few ideas to extend our research, including restreaking our bacterial strains to reconfirm the biochemical properties of our bacteria. The second would be to inoculate brassica seeds with the Achromobacter bacteria, because we found literature on the testing of Achromobacter on Brassica napus, and we wanted to see if this bacteria would have an effect on a different species of this genus. The third experiment is studying the salinity stress in our sweet pea plants with and without the inoculate genus Paraburkholderia, because we found literature on the resistance of Paraburkholderia against salt stress.
We would like to acknowledge these authors for their work. It was beneficial for us to match up our findings with their previous experiments.
We would also like to acknowledge the Loyola Marymount University Biology Department in the Seaver College. Special thank you to Dr. Gasparian, Dr. Lum, and Ava Talui.